We're going to talk more about immunization, immunity testing, vaccinations. So, vaccinations. So vaccine, it's an organism or fraction of an organism that is used to induce active immunity. So do you know where vaccines first started from? Have you heard the story? So Jenner was noticing that the milkmaids were not getting smallpox and found out that cows had cowpox. And so that was making them immune to smallpox. So being the time that it was, he took a volunteer who happened to be an 8-year-old boy and purposefully infected him with cowpox. And then, to make sure it worked, purposefully tried to infect him with smallpox. And when the boy was resistant, we're like, we have vaccines. And so that's where we get the VACA from, because it started with cows. But yeah, it's one of those like, okay, there's some things in the history of science that weren't very ethical. Yeah.
Just prior to Jenner, John Adams, the second president's wife, had a physician actually inoculate herself and her children with cells, they didn't know if they were cells, but basically pus from people who had died of smallpox, not cowpox, but smallpox. And then they came down with the disease, but all survived. Were they just lucky? Or was there something going on there besides luck?
That's a different type of vaccine, because there are multiple types of vaccines. So the idea of the cowpox is that it was a weakened version that our body could deal with, and so we could survive that, but it was close enough to smallpox, our body would recognize and be able to fight it. For the most part, smallpox ends up being too strong for our body to really fight very well. That's why there's a very small percentage that actually do survive. They just have a stronger immune system. But if you take smallpox and you take like the pus or something that's been, that's usually weakened. And so they were taking weakened virus. And so I think it's a live attenuated, what it's called. We'll talk about it more. But that's just a different kind of vaccine. Okay? Yeah. Attenuated. Contains live but weakened microbes. You can also have inactive. And so this is the whole agent contained. You can basically get them, kill them, but they're still there. All you need is that little epitope. And so you can punch holes in the cell wall and make it so that it's not a functioning microbe, but you still have all those parts for your body to recognize as this is foreign, you should mount a defense. So you can either kill the whole thing or you can do a subunit. And so this is where you just can take a piece of it, like, oh well, this is a protein that's on the cell wall. We're just going to manufacture a whole bunch of this protein and just give people the protein. So we build an immunity against that protein, and then when that virus comes with that protein, we're able to fight it. We know what it is, or that microbe. So attenuated. A lot of times they'll weaken them. They'll either grow them for massive quantities of time, a lot of generations, on food sources that they don't really like, so they're not growing very well. Or they'll be old viruses. When a microbe's getting kind of towards the end, it's not quite as virulent sometimes. So it depends on what the microbe is, what you're trying to vaccinate against, which ones they're doing. But yeah, the basic point of the vaccine is to acquire an immunity without actually getting the disease or the disease symptoms. And so usually we're doing these for things that if you've got that disease, it's going to knock you out for a while. We also do the flu, and that knocks you out for, if you get real flu, it can knock you out for a couple, weeks sometimes, but it's one of those things where you're like, okay, how can we acquire immunity without really getting those disease symptoms? So just quick talking about vaccines. Toxoids contains inactive toxins. So sometimes you might want to have just a piece of the toxin that, is the whole point. If you can mount an immune response, that's all you need. We're trying to make sure we do that primary response. So that if we see it again, we have a secondary response much quicker. Another thing they do is they do recombinant. So you can express the antigen gene in a cell. You can then isolate the antigen protein and use that in the vaccine. So this is where you can, shove a piece of that gene for the protein into, say, yeast, let yeast make that antigen, and then all you have to do is purify those little pieces of protein, the antigen, which is an epitope, and now you have something to make antibodies against. Okay? Making sense? Questions? So this will just kind of show you the immunization. They stopped doing some of the immunizations once the people who were dying from the disease was because of the vaccine. Because some vaccines, for some of these, like polio, they were still getting low instances of people actually getting the disease. And so, for the most part, it's like, okay, when you take the vaccine, you have, like a 98% chance you're going to be great and fine, and you're going to be, that's a 2% chance you're really going to get it. And when you're in time in the decades where, most people are coming down and dying, you're kind of like, well, that sounds like a good idea. It's 2% versus I'm probably going to get it. But then when we start getting, into the 1965s and stuff, you start getting better numbers, then you start going, okay, we're going to stop vaccinating because unless there's a good reason, people are fine. They're not getting this, and we don't want to cause problems. And so that's why, like smallpox, we don't get vaccinated for that anymore because they basically eradicated it. So unless you're doing something specific, military or scientific research, we don't get smallpox as a general population. You don't get that vaccine because there's too many people.
Is that because you have a good chance or there's a significant chance of getting it still? Or?
There's still some, but also it's eradicated. The idea is that smallpox should only be in scientific labs. Ideally there is no smallpox roaming around. We've conquered that, that bug. And so the idea is that since it's not out, shouldn't be out in the public, general people shouldn't have to deal with it. Now, if you're a scientist and you're working with it, you kind of want that vaccine because you actually have a chance of getting it. But as a general population, we shouldn't come in contact with it. But you can see just that, introduction of the different vaccines. I mean, for a while, the polio vaccine wasn't that great. It was better than not getting vaccinated, but you can see that there still was, some people getting polio even with the vaccine. Versus once they start making better versions of it, learning what to do, you get a much lower, a drop off of people actually reporting having polio. Measles, once you got that vaccine, drop the numbers. I mean, we all get the MMR, measles, mumps, and rubella. Okay, so the two methods of immunity: active immunity, passive immunity. Okay? So, the active administration of an antigen, so the patient actively mounts a protective immunity response. Okay? So, you are given something, antigen of some sort, so your body will mount an immune response. Versus passive, where an individual acquires the immunity through the transfer of antibodies formed by an immune individual or animal. So this is where the whole breast milk comes in. But also for some of them, they can try to actually just pass on the antibodies. I think they probably more do this in the military because you're trying to make sure that those people, that they're immune to everything. But it's like, if you can just kind of give them the antibodies, okay, here you go, you already have this immunity. So,
is that through like shots or like blood transfusion or?
Usually shots.
Okay.
I mean, the Flonase nasal spray is kind of a new one.
Oh, is that what that is? Because
they can do that for the flu usually.
Okay.
That's misting it, but usually.
So that stuff doesn't contain the virus, it contains just the antibodies?
No, that contains the virus. But
okay.
I think that's one of the few things that has that. Yeah. I think that one's like live attenuated. I'd have to look it up, the changes from year to year, which one, what they're using. They have to make a new flu vaccine every year, so.
Would immunization fit into either one of these, since the immunization causes you to create the antibodies, or doesn't it really fit in either one of these categories?
What do you mean? 'Cause they're
Fits
both
into active.
immunizations. I mean, I'm
No.
understanding your question.
Active immunity is your body's response, making antibodies to antigens that show up, is that correct?
Yeah, but that's your active immunization.
Okay. Well, immunization, I'm sorry, that's right, I said immunity, sorry.
Okay. So, yeah. This is why we give children shots. Yes?
So, active is when it's actually given to you by another person, like a shot or something?
Active is when your body has to mount the primary defense. Passive is when you don't have to personally mount that primary defense because you're given the antibodies. So you get to skip that first step. Because both of them you're being given something. You're either being given the antigen so that you can mount a defense against it, or you're being given antibodies so that you can have that primary. So it just depends on what you're being given. Okay? Types of vaccines. So we're going to talk about live attenuated, inactivated, so live attenuated. So this is where you use pathogens with reduced virulence. It can result in mild infections, but not the true disease. I mean, if you get that flu shot, you might develop a slight cough, kind of not feel great for a day or so. But that's nothing like being flat on your back for days or weeks. An active microbe will stimulate a very strong immune response. So it's one of those things like, because it's alive, it's going to stimulate that response, and that's what you really want. And also, that can provide contact immunity. So that means that if I get it, if I get the shot or whatever with the live virus or the live microbe, then I can actually infect those around me. And so they can end up getting that same level. So it's a contact immunity sometimes. So just because one person in the house has it, maybe the entire household becomes immune to something. They all kind of get that same small bug. Yeah.
What was the very first thing you said? It says use pathogens with reduced what?
Virulence.
Oh.
Reduced ability to make you sick.
How do you
So-
spell that?
Huh?
How do you spell it? Virulence? I wouldn't know.
Because if you think about it, if you have a live microbe growing in you, you're going to mount an immune response, but also you can pass that on. It's weakened, so everyone should have the same weakened microbe, but yeah. So reducing the viability or the virulence of the microbe or the pathogen is called attenuating. So attenuated, that's why it has that name. It's also the most common way, because you just raise it through multiple generations. They start to weaken over time, grow into something they're not adapted to. And so eventually they just kind of lose their ability to really produce the disease. So they're live, they're attenuated, but they're not very virulent. Okay. Inactivated viruses, killing them. Very simple. Usually you use whole agent vaccines. You can also do subunit vaccines with the inactivated. You can do either one. Both of them are safer than live because it's dead. There is no way it's going to really get you sick because it's dead. But it doesn't mount as strong of an immune response as the live ones. You know, your body kind of goes, okay, this was here, I should mount some kind of immune response, but it's not really a threat, versus this is a threat, you know, code red, let's go kill this. Yeah, because, I mean, if you're doing something that's killed, it's not gonna produce very many antigens, it's not gonna really kickstart that immune system. Toxoid vaccines. Okay, these are chemicals or thermally modified toxins. So we've talked about, proteins, chemicals, and I mean, in general, it makes sense. If you heat things up, they're not gonna be as good or as strong or as potent as if they were treated properly. And this is why, in general, when you're handling things, you don't, go and boil things because they tend not to like it. They tend not to work right. It's useful for some bacterial diseases, so that way we don't get the bacterial diseases. It stimulates an antibody-mediated immunity, but a lot of times it requires multiple doses because it has very few antigenic determinants. So there's not much there, so you have to keep getting multiple shots of it. Combination vaccines. So these are vaccines with several antigens, so multiple antigens. They can be from different pathogens. So kind of like, okay, well, we know we have these we want to protect against, let's just throw them all into one tube. So it's nice because it's, you get one shot instead of maybe 3. But several of these combination vaccines have been approved by the CDC. The idea with these is that it helps to make them, or with gene technology, we're trying to make them cheaper, safer, and more effective. So how can we go for something that's like live, but yet it's safer, like the attenuated or the inactive, but yet cheap, and so how do we do this? And so that's where you come into these recombinant gene technology. Because, so if we start taking pieces, throw into yeast or into bacteria, can we make a whole bunch of them? Yeah.
I was writing something, I may have missed something. Would heating up the actual cell or whatever which is producing the disease to the point where the proteins would work be an example of toxoid vaccines?
Well, toxoid is against an actual chemical or toxin. If you're heating it up like that, you've probably just killed your microbe and made an inactive vaccine.
So that would be an inactivated vaccine.
I mean, if you're gonna...
I'm sorry, could you give an example of a toxoid vaccine then?
Because that's usually, it is against some bacteria, but it's usually the chemicals on the bacteria. Tetanus is a toxoid, and diphtheria are toxoid vaccines.
So they're just chemicals that are whipped up in the laboratory?
It's stimulating your immune system against toxins. So we're stimulating against the toxins of tetanus or diphtheria. That's what that whole point is that they produce a chemical that's not good for us, and so we're making an antibody